All right, and our last speaker in this session before the discussion is Silvie Huijben from Arizona State University.
All right, thanks very much, Matt, and thanks for the opportunity to give a talk. Can you hear me in the back? Yeah. All right, so I'm going to talk about insecticide resistance and particularly like what the selective pressures are for insecticide resistance. So first question, of course, why should we worry about insecticide resistance? First of all, it's very widespread. So this is just a map of Africa, Southeast Asia. Every dot there represents when we tested for Anopheles resistance. And every single time when we're looking, we find resistance. So it's clear that we're not looking everywhere, but everywhere where we look, you see a darker dot, that's where there is resistance. So it's widespread. And then for a lot of mosquito-borne diseases, we don't really have any other alternative than vector control. So malaria being the exception, but a lot of other diseases like dengue and Zika, we're mostly relying on vector control because we don't have drugs and we don't have any vaccines. And even in the case where we do have alternatives, so we do have drugs, in the case of malaria, we still find that vector control is our major asset when it comes to reductions of cases. So in this study, what they did is they looked at the reduction of malaria cases from 2000 to 2015. And you can see the red line there is what the actual observed cases are. The top black line is what we would predict if we don't have any kind of intervention at all. And what you can see is that the green part is the part of the reduction that's attributable to insecticide-treated bed nets. So that's when we use bed nets, that's the reduction that's caused by the bed nets. And then the purple part is when we have indoor residual spraying. So again, that's part of reduction. And the middle part, that pink part, is the only part that's can be the reason for the reduction, that's drug treatment. Okay, so even if we have drugs, it is the vector control that's really the main key for reducing caseload. So of course, if we have insecticide resistance, that's a major issue and a major concern. Now, there's lots of different ways how we could then use resistant management to try and reduce the evolution of insecticide resistance. I'm not going to go into all the details, but just really briefly, for instance, we can do mixing of drugs or mixing of insecticides. And we do this a lot for drugs, like it's the commonality, right? So we use combination therapy to prevent resistance evolution, but for some reason, we haven't really translated that much to insecticide resistance control. And then there's mosaic or rotation, so you can change insecticides over space or over time. We can use lower dosages, we can use refugia, so we just not treat part of the population. And of course, the best way is just not use any chemicals at all. Now, what we need to know is which ones of these are working really best, like which ones are best at maintaining insecticide resistance or keeping it under control. Now, to do that, we need to know what the selective pressures are on these resistant mosquitoes. Okay, now there's a lot of gaps in this. So first of all, we need to know what the resistance is or what the susceptibility is at different dosages, because perhaps in contrast to drugs, mosquitoes are exposed to a lot of different dosages because insecticides wane over time and they might not be exposed to the same levels of insecticides. And of course, as we just heard in the last talk, what is the role of fitness costs? Do they have fitness costs? A lot of these resistant management strategies are based on a fitness cost. So if there's no fitness cost, then obviously some of these are not going to work. And then really one critical component that's lacking mostly is our knowledge on heterozygotes. So a lot of resistance evolution, resistant management strategies theory is based on bacteria, which are haploid. The mosquitoes are diploid organisms. So we need to know what's happening with these heterozygotes because they play an important role in resistance evolution. So what our aim was to do is to really figure out what is the level of resistance or what is the level of selection at different dosages. And what you have is at a very high dosages, you have this resistance mutation prevention window. So it's if you go to a high enough dose, you're going to kill everybody, right? Everybody, even if you have a resistant mutation. But then at some point the window opens, and this is when selection for resistance happens. And then at some point that window closes again, and you might even have a window of negative selection, right? So you're going to select maybe against these resistant mutants if there is a fitness cost. So we wanted to test this using Aedes aegypti mosquitoes, and particularly using them as a model system for resistance of mosquitoes that have mutations in the voltage-gated sodium channel. And there's three different mutations at the 410, at the 1016, and the 1534 locus. And this is work mostly done by Rachel and Brooke, two grad students and a collaborator, Eldon and Step at the USDA. All right, so what we did was we created 6 different genotypes. So 6 different lines of mosquitoes. So we started with 3 different parental lines. And these had their mutations. I'm going to focus mostly on the 1016 and the 1534. So these had either VVFF, so that's mostly susceptible, and then VVCC, which is intermediate resistance, and then IICC. So those were homozygous lines. And then we created crosses between those to establish the 6 different lines that we had. And then what we did was we exposed them to insecticides. So the way you do that is we knock them down on ice, and then we use a very a pipetter to just give them very accurate dosages of insecticides. And then we create those response curves. Now, just to walk you through this, because this is kind of a busy graph here. So what you have on the x-axis on the left side is where there's very low levels of insecticides. On the right side is where there's very high levels of insecticides. And then that left curve, the gray one, is Rockefeller strain. So this is a lab strain that's been in the lab literally for almost a century now. So it's very susceptible to insecticides. And as you can see, the dose response curve is very much to the left. So very susceptible. And then we have IICC, they're very much to the right. So these are the most resistant one. And then you have the jumbo in the middle of the different genotypes that we have. And what you can do then is you can calculate the lethal dose that kills 50% of the mosquitoes, so the LD50. And then what we found is that if we either control the locus number 3 or we control for locus number 1, we found that the heterozygotes were mostly falling in the same category as the wild type genotypes. So this is mostly a recessive trait. All right, so then what we did is we exposed these mosquitoes at different temperatures. So we picked just 3 of the different genotypes. So we kept one of the genotypes constant, and then we just looked at homozygous resistant, heterozygote, and homozygous susceptible at different temperatures. So we had 22 degrees, 27 degrees, and 32 degrees. And what we found was that for most of these genotypes, there wasn't any difference in whether they were exposed to high temperatures or low temperatures. But for the susceptible mosquitoes, they actually had higher mortality rate at colder temperatures. So it seems like it's easier to kill mosquitoes when it's cold out than when it's hot out, which for someone who's living in Arizona isn't good news. Okay, so what we found is that there is strong selection for particularly this IICC genotype when there's drugs around or when there's insecticides around. So what about fitness costs? So this is really preliminary data. So we're still collecting this data, but just as a sneak peek, I guess. So when we compare these different genotypes, so far we haven't really found for a lot of the different life history traits, we haven't really found a lot of differences between whether you're homozygous resistant or whether you're homozygous susceptible. They pretty much have the same life history trait results. So number of eggs laid per female, for instance, on the left side, or the hatching success. So how well do these eggs hatch? It's all the same. However, again, we just picked out those 3 different genotypes that I mentioned before. So we have homozygous resistant, heterozygote, homozygous susceptible. And again, we put them at different temperatures. And what we found is, first of all, there is a difference that you can see here. So this is on the y-axis is time to emergence. So basically, how long does it take for larva to develop to an adult mosquito? So what you can see is that there is a big component of temperature. So if you put mosquitoes at a higher temperature, they're going to develop faster. And we know this has been seen many times before. But what we saw is that the IICC, those are these homozygous resistant mosquitoes, they take longer to develop than the heterozygotes or the homozygous susceptible ones. Okay, there's no interaction there. So it's not dependent on temperature. So it goes down at the same rate. Now, something interesting that we found, and this is not significant, so it could be that I'm just staring at this graph and see what I want to see rather than what's happening, is that these Homozygous resistant mosquitoes on the left there, when you put them at a high temperature, they had a lower survival compared to the other ones, but it was flipped for the homozygous susceptible. So homozygous susceptible ones had the lowest mortality at a low temperature, and the homozygous resistant ones had the lowest mortality at a high temperature. And it could be that there is an impact of temperature here, because we know this gene has some heat sensitive aspects to it. So obviously we're repeating this experiment with more replicates to see if this is really what's happening. So, you know what you're showing me, okay. So we know there's a big selective pressure for these mosquitoes, for the IICC mosquitoes. We don't really see a very strong fitness cost. So what we want to know is, well, what's happening really in the field, right? So we can observe these things in the lab, but we want to know what's happening in the field. So this is a map of Phoenix metropolitan area, and every single dot here is a mosquito trap that the Maricopa County vector control sets. And they set it and they empty it and they test it every single week, every single trap. So these are 800 traps. It's a massive data set. It's freely available if anybody is interested in it. And what they do is mostly they test for West Nile virus. So they're mostly collecting Culex mosquitoes. But as a sort of like bycatch, they also collect Aedes aegypti mosquitoes. So what we did is we went back to the mosquitoes, the Aedes mosquitoes that they collected from 2017 up to 2021, and looked at what is a genotype that they found. And then these different gray lines here are just different localities within the Phoenix metropolitan area. And what we can see is that overall this IICC has been increasing, which is what we would predict, right? Because it is mostly recessive traits. So we would expect it, once it's already at a higher frequency, to rapidly go to fixation. And then in some of these areas, it was already fixed in the population. Okay, so in summary, what we know is that there is an incomplete recessive haplotype. Okay, there are weak fitness costs associated, and we've seen an increase of these genotypes. So what we really want to know now is that what is the likelihood of reversion? So can we use, is this model system, or not really this model system, but are these Aedes aegypti mosquitoes likely to revert in the absence of insecticides? Right? Can we use resistant management strategies? So what we're doing now is we've mixed these different genotypes together, and now we're exposing to the different resistant management strategies. So they're all together, and then either they're not receiving any insecticides at all, or they receive a low dose, it's like an LD25 or an LD75, so high dose. Or we have an evolutionary refugee, so only 50% of mosquitoes are exposed. And now we're following them, so this is ongoing. We're sort of like around generation 5 at the moment. So hopefully I'll be able to tell you more about that later. And then we're doing the same thing, but then in silico, because then obviously we can replicate a lot more generations than, you know, about the 10 generations that it takes for 1 year to do this. So with that, I'd like to thank everybody in the lab. Some people were asking where Kijn is. He's the one in the nice little pink band there. This is our 80s party, if you understand the pun. Okay. And thanks to Alden Estep, who works a lot on this, and Abba Gamel, and the funders. Thank you.
Silvie, over here.
You're on the right.
Hi,
On the right
Silvie,
here.
it's Megan. Yeah, thanks for the really nice talk. I was wondering a bit about the cost of resistance in terms of larval competition, like whether you've looked at different densities of larval rearing and whether there's an interaction with temperature, like if the resistant ones are more sensitive to competition.
Okay, so I'll just repeat it. I don't know if it was clear. It was a little bit muffled here, but whether there's a role of larval competition on these outcomes in the life history trait and interaction with resistance. No, but it's our plan to do that. So our next steps is to test for these fitness costs and these life history tables under stress conditions, because, you know, they're always at like optimal conditions. So what we want to do is, yes, under interspecific competition, intraspecific competition, the different temperatures we already started, and just several other stressors. So yeah,
Yeah, cool.
Good question.
Silvie, over here.
Great talk. I'm curious whether you have measured potential costs of in terms of adult survival of resistance, and also whether those individuals that took longer to mature to adulthood, whether they ended up with a bigger body size.
Okay, great. So to answer first your second question, so yes, so the ones that take longer to develop, normally what you'd see is that they are bigger, but we measured their size, both in terms of like dry weight and in wing length, and they're exactly the same. So there's no difference there. And then your first question was, oh, longevity and survival. That's actually currently ongoing. So, you know, they're not dead yet, so I don't have the data.
Hi, I may have missed this in the third section of your talk, but in that trend line that you were showing the annual variation in mortality for the genotypes, do you account for seasonal trends? Is there much variation in the proportion of genotypes depending on whether it's the winter or the summer?
Yeah, that's a really good question. So is there a difference, like, is there a seasonal effect? We haven't looked at that yet, but I think that's a great idea. I mean, there is a lot of reasons why it would, because insecticide applications are very seasonal. So it would be really interesting to see if there is seasonality in these data too. Yeah.
Excellent. So thank you, Sylvie.